Ladies and gentlemen, I have been doing my research work under the supervision of Dr. László Siklós at the Biological Research Center, and our research group is mainly interested in neurodegeneration. In the following minutes, I would like to present our current research regarding to acute neurodegeneration, but before going into details, let me tell you a little bit about its background. Neuronal degeneration is not limited to the degeneration of the neurons because they are all surrounded with non-neuron cells called glial cells. Glial cells can be found in the peripheral and in the central nervous system. In the peripheral nervous system, we can find Schwann cells. In the central nervous system, there are oligodendrocytes, astrocytes, microglial cells, and ependymal cells. Originally, glial cells were believed just to provide structural support for neurons. However, they have more kind of function. The word glia literally means neuronal glue, but as I mentioned, they have much more function. For example, Schwann cells provide myelin sheets for axons and have an important role in regeneration too. Inducing acute nerve injury is an optimal model to study Schwann cells in neuronal degeneration because in this case the axon is surrounded with these Schwann cells which we are interested in. So the process which occurs distal to the injury is called Wallerian degeneration. It is known that due to acute nerve injury there is an increased intracellular calcium level in neurons, but we don't know about what happens in Schwann cells. So that's why we are interested in our research. Thank you. Now you may wonder why calcium is so important. To be honest, calcium is tightly regulated in the human body, and there is an optimal level of calcium when the cells are happy. Neither too low nor too high calcium is good for the cells. An example for too low calcium is muscular atrophy due to immobilization. And in the case of high intracellular calcium, a number of enzymes gets activated and it leads to the death of the cells. So in our research, we wanted to examine the intracellular calcium level in Schwann cells after acute nerve injury. Therefore, we performed sciatic nerve injury, and calcium histochemistry was performed 12 and 24 hours after the injury. Small species of the sciatic and the tibial nerve were removed for electron microscopic calcium detection. In the following slides, I will show you a number of electron microscopic images. But first of all, let me explain what you can see on this slide. First, the axon with green, which is surrounded by the myelin labeled with red, and it is produced by the Schwann cells, as I mentioned, and its nucleus is marked with blue, and its cytoplasm with purple. We measured intracellular calcium in these cytoplasmic area marked with purple. So in higher magnification, you can see pictures from this cytoplasmic area. You can observe some black structures over here, 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 here. These are so-called electron-dense deposits which contain calcium in it. So these are actually calcium-containing precipitates. If you take a better look, you can notice that in the case of nerve injury, there is an elevated intracellular calcium level compared to the control. To move on, In that phase of the degeneration, there are no light microscopic signs of degeneration. However, we observed other structural signs of degeneration which affected mitochondria. From a control sample, here we can see a healthy mitochondria with intact inner membrane structure. But if we compare with the ones from acute nerve injury, we can notice that here the inner membrane was disrupted as well as here. However, here we can observe some intramitochondrial calcium deposits as well. To move on, I would like to share a very novel finding of us, because in microscopy there are things that we look for and some that we find if we are attentive. So our novel finding was these structures called cavolae in Wallerian degeneration, because this area is not yet discovered in Wallerian degeneration, and it is very interesting for us. So we found that in the case of acute nerve injury, these small membrane invaginations contain more calcium than the control. So we plan to further investigate this observation in our further experiments. We quantified intracellular calcium in the cytoplasmic area with a well-known serological method, and as a result, the volume density of calcium could be determined in our samples. But how can we be sure that these black structures really contain calcium in it? To prove it, we used energy dispersive X-ray microanalysis, and due to the interaction of the sample and the electron beam, the chemical content of a point could be determined, and as a result we got a spectrum like this, and the characteristic X-ray properties of calcium could be seen over there. So that's why this is really calcium. And here we can see our results. We found that intracellular calcium significantly increased in the tibial nerve 12 hours after injury, which could still be observed after 24 hours, and significantly increased intracellular calcium level could be detected in the case of sciatic nerve after 24 hours. So to sum up my talk, we proved that there is an intracellular calcium increase in Schwann cells, similarly to motor neurons during Wallerian degeneration. We observed ultrastructural alteration, and we found these structures called cavolae. In our further research, we would like to detect the earliest time point of calcium increase, study the molecular background of calcium change, and the role of cavolae in Wallerian degeneration. At the end of my talk, I would like to thank my mentor, Dr. László Sikló, for his support, all of my colleagues for their help, and last but not least, thank you very much for listening to me, and feel free to ask me. Thank you.